Hello everybody, welcome to another video for Biochem 2. We're going to keep on rolling right into Chapter 10, the Metabolism Overview. This is going to be bringing full circle a lot of the material that we've learned in Biochem 1, as well as some of the stuff that we talked about with the lipid metabolism stuff at the beginning of Biochem 2. So a lot of this is going to be review, so we'll see. There's a lot of kind of just reading involved in this chapter, so we'll try and get through it relatively as quick as we can. Hopefully do it in one video. If not, we'll split it up into two parts. But at the completion of this lecture topic, the student should be able to do the following: describe the basic concepts of metabolism as well as its significance, so why it's important. Know the major nutrient and other essential and non-essential dietary components, so kind of thinking along the lines of what are those essential amino acids, those essential fats, the essential things that we need that we can't produce ourselves, that we need to consume in our diet. That'll tie nicely well into the next chapters, which are all about vitamins. So know the basic metabolic processes and pathways of energy production. Think glycolysis, Krebs cycle, electron transport chain, all of that kind of good stuff, as well as the breaking down, of the fats and the proteins and stuff to put into those cycles. Describe and correlate multiple pathways supporting metabolism of the following: the carbohydrate, lipid, protein, and the nucleic acid metabolism. Describe the role of insulin and glucagon and how these hormones regulate the blood and tissue glucose concentrations in fed versus the fasting situations. A lot of this is going to be kind of review of what we've hit on multiple times before. As well, insulin and glucagon is not going to go away. You're going to see that come up over and over and over in future courses, so make sure you pay attention to that. Finally, demonstrate the fate of major nutrients, those carbohydrates, proteins, and lipids in the fed, fasting, and starved state, kind of connecting with insulin and glucagon. Where are those going to go? Are we going to store them? Are we going to use them? Are we just going to get rid of them? Kind of what's that whole process look like? So talking about metabolism in general, it refers to the sum of all chemical transformations taking place in cell or organism through series of enzyme-catalyzed reactions. That's a big key there. That is enzyme-catalyzed reactions. Through biochemical reactions, the living systems acquire and use free energy for their survival and perform the basic cellular functions. The living organisms also control the flow of this energy through few common metabolites via different metabolic pathways. So kind of this whole process, right? We have the nutrients, we ingest those nutrients, we digest the nutrients, we absorb them through the small intestine, and either we take them and we go and we store them, or we can excrete the stuff that we're not going to use. Remember, if we think about just last chapter when we talked about bilirubin, and bilirubin goes into urobilinogen. Urobilinogen gets excreted through the intestines, and that's what ends up giving the microorganisms react with it, and that ends up giving our feces the brown color. So that's what that's referring to. When we absorb stuff, we can break it down for catabolism. We can either release the energy, or we can take those parts of it. After we break stuff down, we can take those parts and use it to synthesize, to make stuff. That's kind of our general idea of metabolism. So the digested foods are digested by enzymes to produce the smallest molecules. They can be absorbed by the intestinal cells. After the absorption, the cells degrade those molecules into even smaller units, release energy to be utilized for the synthesis of new molecules in our body, and also produce the numerous byproducts, which are further utilized by other metabolic pathways. Any excess foods, such as glucose or fats, can be stored in the form of their respective polymers. So think glycogen or the triglycerides, or stored in the adipose tissue. Respective polymers in special tissues for future use when they're needed to produce energy. Waste materials, metabolites of toxic substances, and drug metabolites are excreted from our body through feces, urine, and sweat, etc. So the significance, so it's a highly coordinated cellular activity, meaning that if one thing kind of messes up in the whole process, it's not going to work properly. So it is well regulated, and we take care, and our body makes sure that we take care of it because it's very, very important that it runs smoothly. Cells require sources of enough free energy for basic cellular functions, the synthesis of new molecules, breaking down molecules for energy, excreting the waste products, and then storing the different molecules for future use. Major nutrients and other essential molecules in our diet are the carbohydrates, the proteins, lipids, and we also have oils, nucleic acids, vitamins, minerals, water, dietary fibers, etc. Again, essential concepts. Like I said, this is a lot of reading, so feel free to, increase the speed of this as we go through this. Metabolism is basically digestion of foods through the breakdown of food molecules to their simplest absorbable forms, and these metabolic pathways produce energy, and also the metabolic pathways are engaged in the synthesis of new complex molecules from the simplest components. Thus, it can be categorized as follows. Catabolism, aka degradation. Again, a lot of this should be review. Breakdown of those complex food molecules to the simplest forms that can be absorbed by the cells. The cells further degrade them to their respective monomeric, meaning the single unit monomeric units, through several biochemical reactions, including enzyme catalyzed reactions, simple oxidation reduction reactions, etc. Also, remember those catalyzed reactions, thinking about those dehydration, hydration type reactions. In this process, kind of taking out water, putting water back in. In this process, energy is generated, which are used to continue several cellular functions, including the next step, the synthetic pathway. Again, catabolic reactions are exergonic, meaning that they release energy because they're breaking stuff down. Breaking bonds releases the chemical energy that's stored in those bonds. So that means that these reactions produce or release free energy, mainly through oxidative degradation of the complex food substances. These free energies drive the next important step, the anabolic reactions, i.e. the pathway that leads to the synthesis of new molecules in our body. Anabolism, the biosynthesis. In this process, simplest monomer units participate to synthesize new complex molecules, such as proteins, carbohydrates, lipids, etc. We take those monomers and we put them together to produce bigger stuff that we need to use in the body. The anabolic reactions are endergonic. That means that when new molecules are synthesized from the simplest monomer units, the energy produced from the exergonic reactions are trapped into these newly formed molecules that are stored, which can be later used in the degradative pathways to release energy. In this way, catabolic and anabolic pathways cooperate and are tightly linked to each other. This is kind of like that graph that we saw way back when we talk about energy, how you have a paired reaction. You have an exothermic reaction that's paired with an exergonic reaction that's paired with an endergonic reaction. So that way they can, the exergonic reaction can produce energy, can release energy, so that then the endergonic reaction can grab that energy and put it to use in order to synthesize stuff. And the exergonic reaction is always going to release more energy than what the endergonic reaction needs, which is why our bodies naturally produce heat. So, as one of the byproducts of breaking bonds is going to be extra energy, which gets released in our bodies as heat. So the anabolic pathways produce some high energy molecules like our ATP, NADH, NADPH, FMNH2, FADH2, thioester bonds, more and more. The hydrolysis or the redox reactions releases the free energies trapped into them. So all of those, the ADP, NAD+, NADP+, FMN, FAD, and the groups containing SH are produced accordingly, respectively. So all of this stuff is review. All of this stuff, it's just kind of going back over these general ideas and concepts that we've seen multiple times. Okay, so metabolic pathways are compartmentalized in the cytosol, in membrane-bound organelles such as the mitochondria, endoplasmic reticulum, Golgi bodies, etc. Also, many metabolic pathways are compartmentalized in different tissues. So it's just saying in different tissues, meaning some happen more in the muscles, the different types of muscles. Some happen more in the liver versus kidneys. Certain metabolic components or things that we need they just happen in different tissues in different ways. In any given metabolic pathway, most reactions are near equilibrium, and they follow the law of mass action, which determines the flow rate or flux of the metabolites. All this is saying is kind of what we always talk about, the rate limiting step, allosteric inhibition, where if you make a specific product, once there's product on that side, it tends to inhibit whatever was coming before that. That's all that's saying. A lot of big words for concept that we've learned many times. In each metabolic pathway, there is at least one reaction, which is far from equilibrium, and reactants accumulate above their equilibrium values. This kind of reaction are known as rate-determining steps, and the enzyme regulates the pathway. The enzyme that regulates the pathway is known as the rate-limiting enzyme. That's always going to be our most important enzyme. These rate-limiting reactions control the flux of the pathway. They control the flow of the pathway. The flux changes only when the rate-limiting enzyme changes its ability to either increase or decrease the reaction rate, which can be controlled by various types of specific activating or inhibiting agents or other metabolic processes. Okay, even more reading. The overview of metabolism. Tropic strategies. The dietary prerequisite of our body to live and continue cellular functions represents its source of metabolic free energy. Simple and complex molecules can be used as the energy source. Metabolic pathways are series of interconnected and branched complex to simple enzymatic reactions that generate specific products. The pathways are connected. There are multiple steps. Things can branch off into different spaces to go into different types of reactions. And we have enzymes that are going to generate the specific products. That's all that's saying. The reactants, intermediates, and products are generally referred to as metabolites. Sometimes reactants, we can call those substrates. And then products, we always refer to those as products. Metabolic pathways take place in specific cellular compartments, and some of them are tissue-specific. Of course, the thermodynamics part of it, we already covered this. That's the endergonic versus exergonic, all that kind of stuff. Breaking stuff down releases heat, building stuff up requires energy to be put in. The regulation, the flux in a biochemical reaction is more or less does not change significantly. So the rate at which these things happen more or less doesn't change a ton. The rates of product formation and breakdown of each metabolic pathway maintains a constant concentration. Reaction rates can be controlled. There's allosteric control, either boosting it, inhibiting it, negative, or encouraging it with positive. So if it's negative, we refer that to as feedback inhibition, or negative feedback. If it's in a positive way, we call that feed forward or positive. There's another one that it is. I don't remember what it is off the top of my head right now, but it's a feed forward reaction. You can also have covalent modification. Again, we learned all about those. When things are phosphorylated, they can be activated by, if you have like a kinase. Whenever things are methylated, a lot of times that's either going to turn something on or off. We've seen a lot of examples of that. By changes in the substrate availability, most often if there's more of the substrate, that's going to push the reaction towards the products. And also genetically, you can be predisposed to certain things. So this is where it's kind of a fine line. I'll kind of throw in a little clinical nugget here. There's a fine line in our world. We talk about diabetes is one of the big ones, diabetes mellitus, especially like type 1, type 2, and how you have, the genetic predisposition, or, some people, especially as they get older, will claim, oh, well, I have diabetes because my parents had diabetes and my grandparents had diabetes, something like that. So when we talk genetically, you can have legitimate genetic issues, where genetically you don't have an enzyme, or genetically your body doesn't produce something that it needs to, and those can be serious diseases. As we kind of get older and we see patients that are older, that's part of our job as medical providers to help people make lifestyle changes. And while genes do play a big role, in who we are and kind of our body type and the things that, how we look, sometimes how we act, that kind of stuff, or how our body overall works, in general, the body is going to respond the same to everything. So I just was speaking with a doctor today. He's a type 1 diabetic, but he does not use any insulin. He completely manages his type 1 diabetes through diet, exercise, and lifestyle modification. And he has for almost 30 years. So this is there's a lot of weight sometimes given to genetics or to certain things like that. But at the end of the day, barring that it's, not a complete deficiency in creation of specific enzymes or specific things, and even in some cases when it is, there's a lot of stuff that you can do. It's not a defining factor. Okay, coming back to biochemistry now. So excess dietary fuels are stored in well-fed conditions. There's well-fed versus starved. So we tend to store in the well-fed conditions. Excess glucose is converted to glycogen. Excess glycogen storage leads to production of fats in the liver and adipose tissue. So if we have too much, we can get the fatty liver. The glucose starts to get stored and converted to fatty chains and stored in adipose tissue. Now, proteins are not stored. However, amino acid oxidation produce precursors of glucose and ketones, which can be converted to glucose and then to glycogen, and eventually fats are produced. So a lot of times, if you eat a very, very high protein diet, remember we talked about that positive nitrogen balance, you can lose a certain degree of protein through the urine, but most of the time your body's going to be taking that protein, breaking it down, and then converting it into the glucose and then into glycogen, into fats. Oxidation of those fuels during fasting conditions yield energy. So we're breaking down when we're fasting, we're building when we are well fed. We have our glucose oxidation. We get that pyruvate in glycolysis, which produces acetyl-CoA. And then I'm not going to read through all of this, just pointing out, again, like I said, we have glycolysis, we have the citric acid cycle, we have oxidative phosphorylation, aka the electron transport chain. In order to create more glucose and to store glucose, we have gluconeogenesis, for making glucose from non-carbohydrate sources. Glycogenesis, making that glycogen. Glycogenolysis, breaking down that glycogen. And of course the pentose phosphate pathway, producing the ribulose phosphate, NADPH as well, producing those nucleic acids. Fatty acid oxidation results in the production of the acetyl-CoA. Amino acid oxidation produces glucogenic and ketogenic intermediates. and finally generates our more acetyl-CoA. We'll go back to our amino acid metabolism. Thus, these oxidative pathways of carbohydrate, protein, and fat catabolism produce a common intermediate, acetyl-CoA, that can be used to synthesize many of the new molecules in our body. And then again, those are all of our high energy compounds. Okay. Continuing, glucose metabolism in the different tissues. Glucose is the major fuel for all of our cells. However, the metabolic pathways for glucose oxidation differ in different cell types depending on physiologic demand, so depending on what's going on in that cell, what their demand is. Liver tissue is a major regulator to maintain glucose homeostasis in our systems. Other tissue utilizes glucose and its oxidized product differently. In this section, we'll discuss about these variation in metabolism in various tissue types. In particular, we'll focus on 4 major tissues: the muscle, the brain, the liver, and then red blood cells. A lot of this is going to be repeat from stuff that you've learned before. So hopefully that's a good review. If there's anything that you don't recognize or that's confusing, 2 options. 1, go back to previous videos in Biochem 2, go back to my other playlist in Biochem 1, and find the corresponding video. Or you can even jump onto like Ninja Nerd or Khan Academy, great resources for those specific things that you need to find. So in the muscle tissue, skeletal muscle tissue and heart tissue completely oxidize glucose. These 2 tissues also can store glucose units as glycogen stores for future use on demand. However, muscle tissue does not release glucose in the circulation, so once it's in the muscle tissue, it doesn't leave. Compared to skeletal muscle tissue, heart muscle tissue has a continuous demand for constant supply of energy needs via glucose oxidation because of the regular contraction to pump and thereby supply blood to tissues. So in comparison to heart muscle tissue, the skeletal muscle tissue has various periods of high or low energy demand depending on the condition. Skeletal muscle tissue can undergo complete oxidation of glucose aerobically via formation of ATP, as well via formation of lactic acid in anaerobic condition, which produces less amounts of energy compared to the aerobic oxidation. All of that should sound very, very familiar. Going from glucose, they're storing it into this glycogen, or going into pyruvate in order to go into our whole Krebs cycle, or going to lactate if we don't have oxygen. Now, liver is the principal tissue that maintains glucose homeostasis. That is keyword meaning blood sugar levels maintained by the liver, as well as control various other metabolic pathways, including the major pathways such as carbohydrate, lipid, and protein metabolism. The liver can completely oxidize glucose to maximize energy production, do gluconeogenesis, store glucose as glycogen, supply carbon precursor molecules for lipid, amino acid, and nucleic acid biosynthesis, regulate detoxification processes and related metabolic pathways, maintenance of glucose homeostasis within the body systems, production of NADPH required for reductive biosynthesis of various important biomolecules, and even more stuff after that. The liver is incredibly important. The liver also gets supply of other metabolic byproducts in various reactions in their other tissues, such as amino acids, lactates, acetyl-CoA, etc., and use them to regenerate glucose via the gluconeogenic pathway. Red blood cells, this will be the one that's newer for us. So red blood cells are completely dependent on glucose supply for their energy needs. Mature red blood cells do not possess mitochondria. So glucose oxidation in red blood cells is incomplete and only produces a small amount of ATP. So essentially, red blood cells can only perform glycolysis because they don't have mitochondria. So they form the pyruvate, which do not enter citric acid cycle pathway. These are the kind of things that you're going to be asked questions about because this is the new material. You'll have a couple of questions about old material, about all this stuff like the liver tissue, skeletal muscle tissue, that stuff, but more than likely the most important questions are going to come from like the red blood cell stuff, the new material. As a result, the pyruvate is converted to lactate and is released into the blood circulation and finally arrives into the liver tissue. Liver tissue can utilize the lactate in the gluconeogenic pathway to produce glucose. Red blood cells does not have any significant glycogen stores. Despite this fact, enzymes catalyzing glycogen synthesis and breakdown have been identified within red blood cells. Glycogen deposition has been described in red blood cells of patients affected with certain types of glycogen storage diseases. Remember, think way back. I believe this was actually last trimester as well. When we look at our glycogen videos that we recorded last try, there are a couple of glycogen storage diseases. But again, don't pay too much attention to that. That was from last trimester, so he's not going to test super specifically on this stuff, or at least he shouldn't. Finally, the brain. The brain tissue, under normal physiologic conditions, the brain tissue is almost completely dependent on glucose for its energy needs. Brain tissue is capable of complete oxidation of glucose via major glucose oxidation pathways, including glycolysis, citric acid cycle, and the respiratory chain. The brain also needs a continuous supply of glucose. For emergency purposes, the brain also stores a small supply of glycogen, which can be degraded to release glucose and provide immediate energy through its oxidative breakdown. In certain situations, like in starvation or some disease conditions like diabetes, if there's a short supply of glucose in the brain, it uses an alternative energy source, ketone bodies, discussed later in this chapter. However, glucose supply, even in very small amounts, is required by brain tissue. There are other specialized cells, although primarily glycolytic, that have relative deficiency of mitochondrial population or limited supply of blood or oxygen. Examples of such are some cells of the cornea, the lens, the retina, including leukocytes, white muscle fibers, cells of the testes, and the renal medulla. Intestinal cells possess the ability to oxidize glucose completely. They also use glutamine as another energy source. These kind of like little random tidbits are things that I could see coming back as test questions. Stuff that places things in that don't have lots of specific blood flow and mitochondrial cells. The fed state conditions. The figure below describes the fates of the metabolites during the fed state, meaning that we just ate. Note that food metabolites are catabolized to produce their smallest units or monomers, further oxidized to release energies, which are then used for synthesis of new molecules, and some excess are stored in specific tissues in their respective storage form until needed. So this is a beautiful picture. This is one that I would probably save, where it tells you and gives you kind of all of the information, how everything is connected, how all of this is connected from the intestines. The glucose carries the carbohydrate, this turns into glucose and goes directly through the bloodstream. The fats are carried by the chylomicrons, and it tells you exactly where they go into adipose tissue. Proteins get broken down to amino acids and where those can go, kind of how all of this is all connected. This is a really, really, really good picture. Again, all of this here, talking more about, just going over this picture in general. Remember, insulin versus glucagon, we need to remember that glucose is used by all of those different tissues. A couple little important facts, like the brain utilizes 60 times more glucose in any tissue. A good chunk of our energy, by like percentage and weight of our body, the brain uses way more, uses a good, very, very large chunk of our total energy, compared to any other tissue. fates of the lipoproteins in the fed state, fate of amino acids in the fed state, all of this. And then here's another really good picture showing us, the glucose and insulin and glucagon when glucose goes low. This is during our fasting. So we have our well-fed picture and we have our fasting picture. So there's the fasting state. Blood glucose peaks up 1 hour after the meal, then decreases to oxidized glucose or converts it to other forms. By 2 hours of meal, the level returns back to 80 to 100 milligrams per deciliter. That's an important number to remember. We want to remember about 100 milligrams per deciliter is our max. Anything higher than that, we start to worry, pre-diabetes. If it's way higher than that, you start to think like diabetic type conditions. Hormonal regulation of blood glucose after meal and during fasting condition is controlled by the negative feedback mechanism. Here's all of our eating fasting. This chart right here, it's been tested on in the past. It will be tested on now. This is super, super important for understanding a lot of the mechanisms of the body. And without this whole process happening, the body does not survive. So a lot of people, a lot of professors as well as boards likes to test a lot on this stuff. And this is going to be more, I would highly recommend coming back in here. I'm going to skip some of this stuff for time's sake. But come back through here, open up the book. I'll leave it here, pause it for a second. Read through some of this and go through as you follow. Most of this material we have seen over multiple times, so I'm not going to spend too much time reading this. Again, the prolonged starvation states. After 30 hours of starvation, gluconeogenesis is the only source of new glucose molecules. Again, we've already talked about gluconeogenesis. Those ketone bodies, that's what we're referring to for the brain. These pictures, review these pictures. And that's it for this chapter. So, like I said, most of this that's all here, you should already know. You can go back and you can read through it. But I think if we can get through some of the Linus learning questions, I think that's going to really tell you everything that you need to know from this chapter. So just a little bit over our 30-minute mark. Looks good. Again, if you have any questions, drop them down in the comments. If you're confused by anything, I highly recommend, jump onto Khan Academy, jump on a Ninja Nerd, jump onto these other things. If you have deeper questions on what this material is as well, go down the rabbit hole a little bit, and enjoy and learning and see kind of what you can get out of all of this. There's a lot here. We talk about insulin, we talk about metabolism, we talk about all these things. There's a lot here. There's many, many, many books written all about this kind of stuff. There's a book called Why We Get Sick that kind of talks all about insulin resistance and insulin and its role in our body and everything. And more recently as well, I wrote a paper on this that they're referring now to Alzheimer's as type 3 diabetes. It hasn't gone through all medical communities that's the case, but there are some very strong connections. That insulin resistance and insulin type issues does play a critical role in the development of some of the neurofibratory tangles and the plaques that you get with Alzheimer's. So keep digging into this kind of stuff. Know this well, so that way in the future when this all comes back, you can have a strong foundation to keep building on it. So thank you for watching and have a great night or day whenever you're watching this.